for our next session. Our next session is titled Advances in Therapeutic Development for CCDS 1. So kicking off this session, I am really pleased to welcome Preeti Vyas to the podium. It's Preeti's first time at this conference. She is a postdoctoral research fellow at Dr. Sujata Kannan's lab at Johns Hopkins University. Dr. Kannan's lab was one of the recipients of the Race for a Cure Award by ACD. Preeti is representing the work that has been pursued in Dr. Kannan's lab today. She has a broad background in pharmacology, drug discovery, and neuroscience, particularly in translational studies. She will be talking to us about the work that's being pursued in Dr. Kannan's lab on novel glucose dendrimers that deliver creatine for CTD.
A very good afternoon, everyone. My name is Preeti, and I work as a postdoc fellow at Dr. Kannan's lab at Johns Hopkins Medicine. And first, I would like to thank ACD for giving me this opportunity to be here and present my work, because this is my first time, and I'm so overwhelmed with the niceness of everyone around here. This is a great community. So today, I am very excited to talk about novel dendrimer nanotherapies. This is a transporter-independent approach that has shown to increase brain creatine levels in creatine knockout mice, the transporter knockout mice. So these are just our disclosures. So GD creatine has already been patented, and the conflict of interest is managed by Johns Hopkins University. Here is a quick roadmap of what I will cover today. So we will start with what the dendrimer nanoplatforms are, because I thought it is important to describe what these nanoparticles are. And then we will walk through some of our clinically validated work with hydroxyl dendrimer, just to show how translatable these therapies are and how safe they are in clinics. And then we spend most of our time on the glucose dendrimer creatine conjugate and what we are seeing in our in vivo and in vitro studies. So dendrimers are hyperbranched polymeric nanoparticles that were first discovered in 1978. And the early generations of these dendrimers, they had like amine terminal at the end. So this tree-like structure that you are seeing, this is how dendrimer looks like in a cartoon for sure. And the terminals that you see around, the initial dendrimer nanotherapies had the amine terminal, because of which it was positively charged, and they were toxic and had off-target side effects. So my mentors at Johns Hopkins, Dr. Kannan and Dr. Sujata, you can see their pictures here. They pioneered a hydroxyl-terminated dendrimer, which was neutrally charged and which had hydroxyl ends all over. So it moved in the body just like a water molecule and did not have any toxic effects. And this has been validated in 7 species and in more than 50 models till now, and several phase 1 and phase 2 clinical trials. So broadly, we work with 2 different types of dendrimers. The first one that you see here is the hydroxyl dendrimer, which our lab has worked for over maybe past 20 years, more than my whole research career. And it targets activated microglia and macrophages, and its uptake is mediated by endocytosis. So these, because they are neutral, they move in the body just like water molecules. And because of the size, they easily cross the blood-brain barrier and penetrate deep tissue parenchyma. And wherever the cells are phagocytic or metabolically injured, those cells pick up these dendrimer. So they are not picked up by the healthy cells, only the dysregulated cells will pick them up. And the second dendrimer that is of interest for today is the glucose dendrimer, and we used it for creatine conjugation. We will talk about it in detail, but first let's quickly look at the clinically validated hydroxyl dendrimer just to show the clinical potential. So let's start with an example which is already in clinic. This is OP101, which is called as HD-NAC, hydroxyl dendrimer conjugated NAC therapy, which was found efficacious in multiple clinical trials. And we started testing it in a rabbit model of cerebral palsy, which is also a model of neurodevelopmental disorders. And you see in the first video that the rabbit is not able to maintain posture. It has a lot of falls and not able to lift the head up. And they also have problems with muscle tone. And our CP rabbit, this is how this model works. And we treat them on PND1, and you can see that this is untreated right now, the second one also, but we treat them on PND1, and after that, we start seeing the improvement with GD-NAC therapy. You can see that this rabbit is hopping now, which is like a typical rabbit behavior. So this is the treated one. In a landmark phase 2 study, where we delivered OP101 with a single intravenous dose, it improved hyperinflammation in COVID-19 patients. Dendrimer NAC significantly reduced the likelihood of ventilation and mortality in comparison to 250-fold higher dose. And it also did not increase the biomarkers in blood for the brain injury. So we did not see any NFL level increase in the blood. So this study showed that dendrimal therapies are safe in patients, they cross blood-brain barrier, and they are cell-targeted, and significantly they improve the therapeutic outcomes in several conditions. Now here comes the second dendrimer that we will focus on today. Building on all the prior work on the glucose dendrimer, we have now designed and patented a novel neuronal targeted dendrimer. So these dendrimer are non-biodegradable, made up of glucose, and which is very safe, and they are picked up by metabolically dysregulated neurons and glia in the brain. It maintains high surface hydroxyl density that I was talking about before, and it releases the drug only intracellularly. So once it gets into the cell at intracellular pH, it releases the drug. So we have prepared dendrimer that are non-cleavable too, but the one that I'm talking about today, for the creatine one, it is a cleavable dendrimer. So it releases creatine inside the cell. And they are very safe, and you can add any kind of payload to it, creatine in this case. And these conjugates localize intracellularly in injured neurons. So I'm just showing example. This is my Caesar model. And also in the model of Rett syndrome, we have seen intracellular localization of these dendrimers. They are not only preferentially taken up by metabolically impaired neurons, but also they show mitochondrial co-localization, leading to higher payload near the sites of ATP production, which we need here. So they also work for oxidative stress regulation and cellular energy homeostasis. So I'm not going to talk much about CTD here, but we may know that the treatment requires increasing brain creatine levels, and the challenges are creatine supplementation, that the creatine transporters are not there. So it doesn't allow creatine to get into the cells and enter the creatine phosphocreatine shuttle. So glucose dendrimer represent a great option here because they get into the cell through alternate pathways. So they do not need creatine transporter to get in. So they can just enter through GLUT-mediated endocytosis because this is made up of glucose. So we are actually tricking the cells thinking that it is glucose, but it is picking up the glucose conjugated creatine here. So just going to see how we prepared this. So I'm not a chemistry person, but our chemistry side of our group, they prepared this conjugate. They first conjugated creatine with the glucose dendrimer using click chemistry, and it is very pure. They have characterized it using MALDI mass spec, and they have quantified the size. So this is very well characterized and very stable. And it releases around 80% of creatine in 7 days under the intracellular conditions. So in this slide, we just show that it enters the mouse hippocampal neurons in vitro. These are the healthy cells, so not the diseased one. So that's why the most important question we asked was, what if there are no transporters, or we block the transporters, then what happens? So you can see in these graphs that hippocampal neurons treated with free or glucose dendrimer conjugated creatine, their entry into the cell is different when you block the creatine transporters. So we blocked the creatine transporters here using 3-GPA, which is a creatine transporter blocker, and we observed that the level of uptake once the transporter is blocked is changing with free creatine but not with GD creatine. So it doesn't matter whether the transporters are active or not, but it will still be picked up by the cells. We also observed that GD creatine was more efficacious than free creatine in preventing excitotoxic and oxidative injury. Now, we did it on mouse hippocampal neurons from healthy mice, but it was an important question that how they will localize in patient cells. And we cultured the fibroblasts from the patients, CTD patients, and we observed the internalization and perinuclear uptake of GD creatine in the patient-derived fibroblast. Right now, we are continuing these studies in both patient-derived fibroblasts and iPSCs, and building upon efficacy data. So I'm planning to measure the creatine levels and ATP production and do more studies with iPSCs and fibroblasts. So we have these from the patients. So this is our initial experimental design. I know this looks complicated, but I will simply explain this, that we are treating the knockout mice either with free creatine or GD creatine or saline, and comparing it to wild-type littermates. So we are measuring the grip strength longitudinally. So every week we do the grip strength test, and we also do the weight measurements. And we are also looking at creatine cyst and phosphocreatine cyst, the MRI data longitudinally. So we did a baseline and 3 weeks after treatment, and we are planning to keep repeating it till the end of the study. And after we euthanize, we measure the brain creatine and phosphocreatine levels using MALDI, which is matrix-assisted desorption ionization technique. And also, we measure the creatine levels in the brain cells by using just basic creatine assay. And because we think that we need to measure some secondary biomarkers also, we are also looking at the mitochondrial stress using the seahorse assay. So this was an important question for me, that when to treat, which age. So we did like a therapy at PND 21, and then we also had one cohort that we started treating at PND 54. But I'm still optimizing like what age I should pick, or even should I start treating them earlier than PND 21. So we observed increase in body weight and grip strength in these knockout mice treated with GD creatine, while the saline treated knockouts, they were not catching up. And similarly, as I was talking, that we are also trying the delayed therapy. We saw the similar improvement, but I need to add more numbers to this, this cohort. So I need to treat more mice at this age, and then maybe that data will look better. So we next conducted a pilot study, and we were trying to obtain a longitudinal quantification of creatine and its utilization in cells. And we measured creatine levels and its conversion to phosphocreatine using MRI CEST imaging. So at Johns Hopkins Radiology, we have a group. They have developed creatine CEST and phosphocreatine CEST. So we are collaborating with them and measuring these in the mice. So we have a longitudinal measurement. So we used 11.7 Tesla MRI, where the creatine knockout mice treated with GD creatine, they show decreased level of creatine in comparison to their baseline. However, we observed an increase in phosphocreatine level, suggesting that there is a conversion of creatine to phosphocreatine in the brain. So it's going in the shuttle. And when I looked at this data for the first time, because I am not a person who is involved in MRI and CEST, my question was, oh, look at the ratio. Look at the y-axis. Why the y-axis for phosphocreatine is so low? So then I was explained that, okay, phosphocreatine, it has 2 exchangeable protons, whereas creatine has 4. So it will always be a ratio of 1:2. So creatine will always be double of phosphocreatine even at the same concentration using CEST. So, and we are also trying to look at some relative changes, but I wanted to highlight it here. Okay, we also wanted to measure post-mortem brain creatine levels using MALDI. And we have a MALDI core at Hopkins that we are collaborating with. And we imaged a different mouse. We imaged a different mouse that received a high dose of GD creatine, 100 mg per kg. So to highlight that the efficacy data that I'm showing, it is 20 mg per kg only twice weekly. So I'm dosing twice weekly. And here we imaged a mouse with a single dose of GD creatine, and we collected the tissue after 24 hours and did the MALDI imaging. And we observed a higher level of creatine in the treated mouse. And because this is MALDI, so we are doing a section-based quantification for the unbiased results. And we saw that white matter area show higher concentration of creatine. And also, we did the quantification in the whole brain and saw the improvement there. Once we knew that GD delivered intracellular creatine independent of creatine transporters, we now wanted to actually quantify the levels. And we injected 100 mg per kg of GD creatine and collected the tissue 7 days later. So the release profile of GD creatine, I have shown it on the chemistry slide, but within 7 days, there is at least more than 80% of release of creatine from the GD molecule. So that's why I decided that, okay, I want to collect it 7 days after and see how much is released. And we can observe that creatine knockout mice has really low levels of creatine in brain, muscle, and heart. And the treated mouse, however, showed the higher levels. And also because we know that GD creatine cleaves in intracellular pH, we also cleaved the remaining creatine from GD using esterases. So this is a total of what was released before plus what we released after. So whatever was taken inside the cell. To confirm that conjugate is going to areas of pathology in all the models, we tested the localization in our rabbit model that I showed initially, because they also have like decreased muscular strength and the tone is reduced. And we observed that it is localized intracellularly in periventricular region of newborn rabbits with cerebral palsy. So this PVR region is the main area of pathology in our cerebral palsy model. And we have seen the localization, and it also improved the neurobehavior within 72 hours of birth. So I'm also trying to figure out how to check the neurobehavior in creatine deficient mice, because our rabbit scale is very quantified. We know how we can score the improvement. So I'm trying to figure out something similar for the creatine knockout mice too. So to bring everything together, we have shown that GD creatine can bypass the defective creatine transporters and deliver creatine intracellularly. In our knockout mice, we observed 20-fold increase in brain creatine level with 1 dose. And this was not just in brain, but also we measured it in muscle and heart. And the conjugate itself is highly characterized, and it has a very good drug loading. And we saw that there was improvement in grip strength, and the phenotype was improving, and localization in the patient cells, so the payload is actually going where it needs to go. So that was very encouraging for us from a translational standpoint. So the key takeaway is that GD provides a transporter-independent route for delivering creatine to neurons, muscles, and heart. So this is an ongoing work, and we are building upon it. And I would like to acknowledge all the beautiful people that made this work possible, as well as our funding sources and ACD for providing us funding to continue this work. And with that, if there are any questions, I would be happy to answer.
Hi, great talk. I'm really interested in, so you say this is an endocytosed, so it goes into endosomes?
We think it is GLUT-mediated endocytosis, but we are still, because this is comparatively a novel dendrimer, we are still trying to figure out the mechanism. But till now, whatever mechanistic stuff we have done, we think that it is GLUT-mediated endocytosis.
So then how does it escape from the endosome?
I am looking at the mechanism. And in other studies, we have also seen that some conjugates, they require surface transporters, but some are just like go inside through pinocytosis and endocytosis. But this is a good question that I need to really figure out the mechanism of this.
Yeah, it's just a little bit of mechanistic pharmacology that could maybe help optimize the system moving forward.
Yeah, that's good. Thank you for the question.
Hi, thank you for the talk. It was really great to see that brain muscle are restored for the creatine level. Did you see any toxicities related to the level of the creatine that was in the brain? Because it was very high, like much higher than the knockout. And also, like are there any, like how is the dendrimers cleared from the body if it's continuously treated? And then I guess I saw that one of the graphs, the wild type had less grip strength than the treated mice. How is that possible for the?
Yeah, so this is very interesting thing about dendrimers, that if there is an injury, the cells pick them up, and if there is no injury, it goes through the regular metabolism, and it is very inert, and it is cleared by kidney within like 72 hours. And this is also like a clinical data which is also published, that we have not encountered, at least till now, we have not encountered any toxicity with it. But because of its high hydroxyl surface density, it is very inert, and we did not see at least any toxicity till now. And it just clears through kidney within 72 hours. Whatever is not picked up by injured cells is cleared through kidney. So we have done an extensive pharmacokinetic study, and this is what we have observed. But what I'm still thinking to do, like a dose-dependent study in these mice and see if we see any toxicity. But I don't think we will.
Wonderful presentation. Now, these glucose dendrimers, are these like polymer nanoparticles?
Yeah,
Okay.
yeah. These are nanoparticles, but the core is glucose here.
And the nanoparticles, how are you giving them? Orally, IV?
So this is subcutaneously, because I wanted to go closer to the translational
Route?
point. But we have seen that we have injected it in several routes. But for this work, I'm using sub-Q.
Very good. Thank you.
Hi, I have a question related to some downstream processes. So certainly, for any new therapeutic, there's always, it's very important to compare that to the current standard of care. So maybe I missed it, but what I couldn't see is whether or not the changes that you see in creatine levels, how does that compare with if you simply administer a bolus of creatine, an appropriate bolus of creatine to the mice? So is there a benefit relative to just supplementation with creatine?
So whatever we have seen in the past is that you can achieve similar level of efficacy at very low dose of dendrimer. And I think it's a good idea. We should definitely try like a big bolus of creatine and then see what is the difference. But our other models and our all preclinical studies have shown that even if we inject like a high dose of a dendrimer therapy or a lower dose, it will only be picked up by the cells that are needed. So the remaining will be cleared by kidney intact anyways. So for comparison, I'm using free creatine as a control, but I have never tried like a bolus dose. So I think I'm gonna try it to give like a high dose of creatine and then see if what is the difference. But I'm injecting similar dose of creatine as GD creatine, so 20 mg per kg for both. And we are observing that creatine-treated mice are not catching up. And I'm not showing that data here because we were doing it just last week, but now we are also doing the CEST on creatine-treated and the GD creatine-treated and knockout. So we are including all the groups. So the data I showed here is like very pilot data. So I'll do it for every animal. Thank you so much.
Our next speaker is Alex Edwin. Alex graduated with a bachelor's in neuroscience in Santa Clara University in 2023. Since 2021, however, he's worked as a research professional in the Montine lab at Stanford School of Medicine, where he focuses on developing small molecule therapeutics for creatine transporter deficiency. He has used molecular modeling to design novel creatine prodrugs, developed high-throughput fluorometric in vitro assays, and expanded his work to include both in vivo and behavioral characterizations of the SLC6A8 knockout mouse models. Alex is a current ACD fellow as well as the recipient of the Race for a Cure award.
All righty. Good afternoon, everyone. Thanks for spending some time with me this afternoon. Try to make it worth your while. I'm a researcher with the Montine lab at Stanford University in the Department of Pathology. So today I'll be taking you through some of the work we've been doing in small molecule drug development for CTD. Let's figure this out. Okay, so I can walk a little bit with this mic. It's kind of fun. Today we'll hit on a couple of different points. First, we'll talk about the creatine prodrug strategy. What even is a creatine prodrug? What's a prodrug? How did we go about designing them and testing them in cells and bringing them to a point where we could decide which drugs to move to an animal study? Then I'll talk about our animal study outline and how we went about testing the two drugs that went into a long-term examination. Those two drugs I'll refer to as STF-8227 and 9968. So we'll go through the results of those. And then we'll end with some exciting forward-looking research where our lab is starting to move towards. I find it very exciting. I hope I can share that with you today, sharing the excitement. So let's get started. Okay, I thought I'd give everyone a jump scare with a bunch of chemistry, maybe wake us all up after the lunch lull. So bear with me. But this is important to sort of set the scene for the rest of the talk. So let's ask the question first, what is a prodrug? So a prodrug is sort of a modified version of your active drug of interest, and these modifications are made to enable some sort of new ability to the molecule that you're trying to work with. So in our case, it's creatine. Creatine, as we all know, without a functional transporter, cannot pass through cell membranes to function as it would. And so in creatine transporter deficiency, creatine is unable to pass the cell membrane. And so in our case, these prodrug structural modifications are intended to enable this new ability of the creatine molecule to bypass the cell membrane, or bypass the creatine transporter rather, by just passively diffusing through the membrane. So I like to explain it. Creatine's a very polar, very charged, water-loving molecule. It dissolves very easily, as I'm sure parents know, into water. Whereas this cellular membrane is very greasy, kind of like oil. And we all know oil and water don't mix, so that's why creatine can't pass through. And so these prodrug modifications attempt to sort of increase the greasiness of the prodrug molecule, makes it more oily and easier to pass through. And so all these squiggly lines, if you don't remember from your organic chemistry classes, represent carbons, and carbons add a lot of that fat-loving nature. So these will pass through the cell, and they're designed as such that once getting inside the cell, there should be some chemical or enzymatic mechanism that'll remove those structures off of the creatine molecule and therefore release free creatine to act as it would. And so there's 3 different categories that we kind of have been studying in our work. There's the creatine amides, which are off to the left. Then we have our amide esters and our macrocycles. And they're named as such because if you look over here, just a quick chemistry lesson, I promise it'll be short. These 3 nitrogens create a functional group called guanidine, and making an attachment here forms an amide bond. So we refer to it as a creatine amide. Over here we have same structure, but now on this sort of oxygen-containing functional group called a carboxylic acid, we attach on to this oxygen to form an ester, hence the amide ester. The macrocycle just sort of cyclizes this amide ester. So these are 3 different design strategies. Okay, so we've covered the sort of background. How did we go about testing them in a somewhat rapid manner? So our lab designed and evaluated over 75 different creatine prodrugs and tested them in creatine transporter knockout cells, specifically a human haploid cell line. And so we would incubate creatine prodrugs, or creatine for that matter, with these knockout cells for 4 hours at 100 micromolar, and then collect the cells and inspect how much creatine got into the cells from these prodrugs, which for just to promote my poster later, this will be explained in much greater detail on the poster. And so what we found from this screen were 5 creatine prodrugs that were able to deliver creatine to these HAP1 knockout cells under the conditions I've explained. So to orient you to the graph, I'm sorry, I'm swinging from this way to this way, keeping you guys on your toes. The blue bars are the wild type cells, and these orange bars are the creatine transporter knockout cells. And so we can see y-axis being creatine concentration, that these wild type cells have a lot more creatine than these knockout cells. And when you add creatine exogenously, the intracellular contents go up. So that validates that the functional creatine transporter is present. That does not happen when you add creatine to these knockouts, so it's validating the model. But you see when we add these 5 prodrugs, we're able to increase the creatine concentration in these cells without a transporter. And so this confirms previous findings that the amides, specifically, if you think of the 3 categories, the amides, not the amide esters or the macrocycles, are the ones that can deliver creatine in these cell systems, given that all 5 of these prodrugs are amides. So with this knowledge in hand, we took the leading prodrug from that screen, A227, and went through this animal study. So it's broken down into a couple of different phases, so just bear with me. First, what we wanted to understand is what dose should we be using for these prodrugs in a long-term study. So the first phase, day over day, we'll give the mouse an escalating dose of the drug until we reach a point where we start seeing side effects in the mouse. So once we see that the first sign of side effects, we pull the dose back 20%, and that's referred to as the maximum tolerated dose. Then we'll give that dose for 5 days, asking the question, can they tolerate this dose upon repeated exposure now? If that holds true, then we begin the study first by doing a pharmacokinetic analysis where we'll give the drug and then 2 hours later collect tissues that we're interested in, which to name are muscle, cortex, and plasma, and inspect the concentration of creatine in those tissues to see if these prodrugs are delivering creatine as we would hope. If that's the case, then we go into this longer-term study, 10 weeks, where over the course of this 10 weeks, we're sort of instituting this behavioral battery to answer questions related to, are these mice improving in their behavior? Are they improving in their strength? And so our behavioral battery consisted of the activity chamber measuring locomotion of the mice, knowing that the knockouts typically have this hyperactivity trend. We also have the inverted grid test and grip strength meter, which measure muscle strength, and the Y-maze and novel object recognition, which both measure short-term and long-term memory, respectively, in our case. In the last week of the study, we switch into a deuterated version of the prodrug, which means it's labeled so we can trace it as compared to the free floating creatine in your body already, so we can quantify how much drug is releasing creatine after 7 days of exposure, and if there's an accumulation of creatine over time from administration of the drug. Okay, so let's go into some of the results. So each slide will start with a question. So A227, how much creatine does it deliver in a single dose? So we had to figure out again how to determine this dose. So in doing so, if you remember that first little mouse figurine, we escalated the dose until we saw the lowest observable adverse effect dose, a total mouthful. That was 50 milligrams per kilogram when given IP. And so we toned it back 20% and determined 40 mg per kg was the appropriate dose to give. 1/4 of that as standard protocol for an IV dose to get a second route of administration, and tissues collected 2 hours after that dose. And what we saw was that when dosed IP, there was creatine that was delivered into all 3 tissue types in a manner that you would kind of anticipate. Most of the creatine staying in the periphery, with the most being in the plasma, then about 740 nanograms per gram in muscle, less than that in cortex, around 72 nanograms per gram. When dosed IV, though, there was no creatine delivery to the cortex. Kind of interesting, and we're still not entirely sure why that's the case. And a little bit less in muscle as compared to when dosed IP. But the important caveat of this all was that it delivered to all 3 tissue types, which for us was the go, no-go decision in going into a long-term study. So that's what we did. So now then, did it improve behavior strength in the CTD mice? We dosed at 40 milligrams per kilogram. But unfortunately, what we saw in this study was a vehicle-related toxicity. So again, this drug was a creatine amide, and one of the sort of drawbacks of adding greasiness to these molecules is that it makes them harder to dissolve. And so as a result, we had to use a fairly aggressive formulation, which in our 5-day analysis of tolerability, we saw no side effects or no adverse events. But we did see, once we started the long-term study, that mice started to die in around the 2nd week of the study. And this was irrespective of treatment groups, which led us to believe it was vehicle-related. So as a result, we were only able to collect, not too much behavioral data, really the baseline and the 2nd week, which were activity chamber and inverted grid test data, respectively. And what we saw was nothing remarkable. We did see the hyperactivity phenotype from the knockout mice. We did not see a treatment-based effect. And we also saw the genotype effect of strength in that these knockout mice are weaker, but there was not enough time, and we started having this confound introduced to see if this drug would have a meaningful improvement on any of these behavioral readouts. And so we moved then the deuterated portion of the study up so that we could still salvage that to answer the question of whether or not it accumulates upon repeated dosing. And so what we saw was that in the tissues of interest, cortex and muscle, they did accumulate, which is encouraging. We saw about a 9-fold increase in the creatine, in cortical creatine levels, and around a 2-fold increase in muscle. We saw around a 2-fold decrease in plasma creatine, which we haven't come up with a full explanation of yet. Perhaps optimistically, the drug's getting absorbed better. But nevertheless, I think what's important for us is that it's accumulating in these muscles, these high energy demanding tissues. Okay, so after the end of the study, we collected the tissue and weighed all of the different organs to see if A227 affected organ weights. Generally, no, it did not affect organ weights. We saw the genotype effect emerging in several different tissues, but we were kind of plagued with a lower sample size as mice started to die, and so statistical significance and power started to go down. and the age of these mice was younger than we anticipated as the treatments were terminated early. So some of the genotype effects in organ weights had not quite emerged, seemingly. So then we'll move to 9968. Again, same setup. How much creatine does 9968 deliver in a single dose? And so just to set up the stage here, 9968 is a macrocycle. So 8227 is a linear amide, and we were intrigued to see whether the macrocyclic derivative of this compound could behave any better than 8227. So for the dose determination then, it was much easier this time. We wanted to stay consistent and compare to 8227, so we used the same doses, 40 milligrams per kilogram IP, 10 IV, and the tissues again collected 2 hours after the last dose. And so to orient you guys to the graph, we've got the IP data in blue, the IV data in orange. This is 8227 data for comparison. But we had some very interesting findings here. As compared to 8227 when dosed IP, 9968 delivers around 17-fold more creatine to cortex, around 1,200 nanograms. And when dosed IV, it's about twice that. Interestingly as well, 9968 delivered around 9-fold lower or less creatine than 8227 to muscle, suggesting kind of an interesting biodistribution. So that's what we wanted to start to look at here. How does it distribute after a single dose? So now what we're looking at is concentration ratios. What concentration of creatine is delivered from 9968 in, for example, on the left, cortex as compared to plasma, middle, cortex to muscle, muscle to plasma. And we see 9968 dosed IP and IV here. These are the 8227 data, which are barely visible, if not at all, quantifiable. And we see something quite intriguing. For 9968, when you dose IV, you see this very impressive CNS partitioning where the concentration in cortex as related to the plasma in this single dose, 2 hours collected post-dose, around a 40-to-1 ratio, cortex to plasma, which is strikingly high. And so we presume that there must be some sort of brain targeting mechanism of this prodrug, although we have not identified it. And so that's something that is interesting and we would like to pursue a better understanding of that in the future. Okay, so then we move on then to the behavior in the long-term study. Again, we deliver to all 3 tissue types. That's our go, no-go decision to warrant a 10-week analysis. And through our MTD analysis, maximum tolerated dose analysis, we determined 100 milligrams per kilogram was a safe dose to give to the mice. But unfortunately, what we saw was that there was not much of a behavioral effect. So just to walk through. On the left-hand side, we have body weight throughout the course of the study. The blue being the wild-type vehicle. You can see the legend here. Knockout vehicle in red, drug-treated in green. And there was not much of a change in the body weight in the drug-treated mice. There was this mild, although non-significant, attenuation of the hyperactivity phenotype in the drug-treated mice. This over here is like a heat plot. So when these mice are running around in their chambers and their cameras tracking their movements, this is a sort of a plot of the movement in the first 2 minutes. It's just kind of a visual representation to show that hyperactivity and that the knockouts are moving quite a bit more than the wild type. On the bottom here, we have the strength readouts. So on the left-hand side, we have inverted grid test data, right-hand side grip strength meter. And we again see a genotype effect where the wild type show much more strength than the knockouts. But unfortunately, we don't see a treatment effect in these mice. We then start to look at the short-term and long-term memory. We see the genotype effect in the wild type and knockout mice, being that the wild-type mice have an alternation index of around 60%, suggesting that they are not cognitively impaired, but the creatine transporter knockout mice are right around this chance, 50% in both weeks 4 and weeks 8 of the study. Although there seems to be some slight rescue in this Y-maze data, when looking at statistics and incorporating the longitudinal analysis, it's non-significant. Then again, we look at the novel object recognition. This is a long-term assessment where the novel object is introduced to the mice 24 hours after the habituation phase. And in fact, in the skeleton mouse model, there is no genotype effect. So we don't see any long-term memory phenotype in these knockout mice as compared to the wild type. They all have a similar discrimination index, looking at the novel object about the same amount of time. And so therefore, there was no way to see a treatment effect. But we do see this hyperactivity phenotype when looking at the distance that they moved during this time. So then, after collecting the tissue from this study, we ask the question, does the dosing affect the organ weights? And I highlight these particular bar graphs in red just to direct your attention that there was this significant genotype effect in brain weight. The knockout mice have significantly lighter brains than the knockout mice, which is not repaired with the drug treatment. This was not seen in the earlier study, 8227. Again, it could be because it's underpowered from mice dying, or an age-related explanation. These mice were 5 weeks older, and it may be that this emerges later on in the mouse's life. Interestingly, though, we did have an organ weight change in liver and kidney in the treatment groups. You see a significant increase in liver weights and kidney weights. And so we don't know exactly what this is caused by, but we're currently doing histology on these tissues, as well as analysis of the deuterated creatine from the 7-day pharmacokinetic study to see if there's higher creatine levels in these tissues that may explain the increase in organ weight, or if it's due to something else. Okay, so now forward looking. This work has been kind of the summation of the last 2 years of mine in the Muntean lab. This is kind of something that we're excited to be working on for the next year with the help of the ACD grant. So this is sort of a new class of drugs where we've now designed this novel class as candidates for improved therapeutic delivery of creatine to knock out cell types. And so we've begun synthesis of a library of these candidates and confirmed their ability to get into cells with flow cytometry because we're able to fluorescein label these prodrugs. And so we've synthesized 9 of these prodrugs so far, and comparing to a control that's very well established and has been used in other indications in clinical trials, we have so far 5 out of 9 of our compounds are able to achieve equal or superior cellular delivery relative to that known control. And so now that we've confirmed that these prodrugs can get into cells, the next question is, do they release creatine? And so we're now scaling up deuterated versions of these prodrugs to look at that creatine release and then start some early in vivo PK, biodistribution, and behavioral studies, a lot of the things that we went through for the other prodrugs. So to conclude then, we have creatine prodrugs that effectively restore creatine intracellularly in our HAP1 creatine transporter knockout models. STF8227, which is our best in vitro candidate, accumulates creatine in the cortex and muscle with 7 days of dosing, but the long-term studies were halted from the vehicle-related toxicity. 9968, which is a macrocyclic derivative of 8227, delivers roughly 17 times more creatine to the cortex than 8227, with a 40-to-1 cortex to plasma ratio suggesting brain-targeted delivery. But despite this, long-term 9968 dosing does not show convincing improvement of the creatine transporter knockout behavior or strength. And so I'll just interject within the slides here really quick to say, one of the things that even we talked about this morning in the fellowship meeting was it seems that it is still unknown what is the therapeutic amount of creatine to get into brain. What is sort of the floor, if you will, of creatine that needs to be delivered to start to see a change in the behavior, to start to see a change in strength and these sorts of things. And so although 9968 is a great improvement over 8227, it still seems that it's maybe below the floor in an ability to improve behavior and strength. And so it's a little bit unfortunate in that sense, but it is also encouraging data because we do need to determine this floor. It's important for the therapeutic development to understand what we need to shoot for. And so what's kind of encouraging about this, I will say, is if there is this brain-targeted mechanism and that can be characterized, then drugs can be designed to target and exploit that brain-targeting mechanism in a way that can increase the delivery into brain. And so there is a through path here. That being said, we are also examining this sort of next generation of creatine prodrugs in parallel. And so there's a lot of, we're kind of hedging our bets, as you will, to see what comes out on top. And so it's a really exciting time for us. And with that, I'd like to acknowledge my lab. I am 3 years out of undergrad. I have not started a PhD. My applications to med school are sitting on my desk in the hotel room. And so I have a long ways to go, and I would have accomplished nothing without everybody in my lab and without everyone in this room. I came here in 2024 for the symposium, and it's been a massive reason as to why I didn't ship off to medical school sooner. It's been such a fulfilling experience getting to know you all and to do this research. So with that, I welcome any questions.
Thank you. I enjoyed all the chemistry in there. So I'm just wondering if anyone's mapped out the kind of biodistribution of creatine, whether it's isotopically labeled or not, in non-human animals all the way through humans. I know there's been some studies with deuterated creatine, or probably a lot in humans, but I guess what I'm asking is, does anyone know if I give a gram of creatine, how it's distributed in the body or in these models? That's my first question. And secondly, I just want to know if you thought about isotopically labeling the prodrug moieties themselves. You might make them a little more greasier and get them to your target.
Oh, that's interesting. To your second question, I've not thought about that, but would love to talk to you about that later. To your first question, I don't know offhand if people have isotopically labeled creatine and looked at biodistribution. I wouldn't be surprised if that data existed. That would also be a fairly easy experiment for me to do on my own. It might be a good idea to give a sort of positive control to see the biodistribution of natural free creatine versus a prodrug and see how much they differ. That would be interesting.
Thanks for a really interesting talk. As an outsider, as I was looking at your data, it seemed to me that one way to explain the absence of effects due to creatine, even though it appears you have delivered a lot of creatine, may be related to how you actually detect the creatine. And so do you know, when you say that you've delivered a certain amount of creatine, do you know that it's creatine the molecule, or do you know that it's the prodrug or some intermediate there in between? Because you could imagine that if you're delivering the prodrug and the prodrug isn't being converted into creatine, that could explain why you're not seeing any behavioral effects.
Yes, so I'd love to have you come by my poster later. But to answer your question, the power of isotopically labeling these molecules is that you can distinguish between the prodrug and the free creatine. So when I show those data in the animals, we are confident that it is free creatine from the mass spec methods. That being said, we have not measured the prodrug concentrations in brain. So we're looking at only part of the story, to be fair. We're only looking at that creatine which has been liberated from the prodrug moiety in these tissues, but we have not looked at the uncleaved, unhydrolyzed, intact prodrug concentrations in brain. That would build out a better story. The only reason we haven't done it is cost related. Running all of these samples through mass spec is pretty expensive, and you would have to develop novel methods to detecting each of these prodrugs through mass spec, which is pretty costly. But we're certainly open to it, of course. No.
Just out of curiosity, I mean, you can measure obviously the one that is deuterated, that it is, only a few units of mass away from the regular one. How much of the total amount of creatine can you change with the deuterated compound? So in other words, how much it is endogenous creatine and how much is the creatine that you give with the prodrug?
So We have not done mass spec of non-deuterated and deuterated prodrug in these samples together, but that would be a good idea to get a clearer and bigger picture of what relative amounts we're delivering. Yeah. And it would be something that's easy to do because those methods are already established for non-deuterated creatine detection.
Sorry, one other suggestion is that obviously if the creatine is taken in and utilized, it would be converted into phosphocreatine. It would be nice to get phosphocreatine as well, both with the endogenous one and the deuterated one.
It's a great suggestion. I think that would, yeah, and I think best case scenario for our work is we'd be able to detect intact prodrug, deuterated creatine, deuterated phosphocreatine, and non-labeled creatine, non-labeled phosphocreatine. That would get the entire story. Yeah, the cost just goes up. So, trade-offs, I suppose. Thank you.
Our next speaker in this session will be Dr. Xin-Yi Chen. Dr. Chen is a molecular neuroscientist at the Frolin Biomedical Research at Virginia Tech. She has conducted studies on multiple rare diseases with a focus on epilepsy before her work brought her to creatine transporter deficiencies. Her current research explores the application of focused ultrasound as a therapeutic approach for CTD to enable better delivery of drugs across the blood-brain barrier. Dr. Chen is a second-year fellow at the ACD Fellows Program, and so her presentation today covers much of the work that she's done over the 2-year fellowship. Dr. Chen.
Thank you, everyone. Oh, the talk is very exciting today. So many new drugs make me feel so exciting. That's the new hope here. So today I'm here to introduce, is this the monitor? So here we are going to introduce how I can change the slides. Sorry, this one. Okay, so here is our lab. We are doing a device to penetrate the blood-brain barrier and make the limitation of any drug that have a limitation to go through the brain. They can have beneficial from our technician by the focused ultrasound. We are new lab just set up at the Virginia Tech on 2024. So we have 2 branches, one at the Virginia Tech, another one is at the Children's National Hospital. So here is our team. At the first one is our physical scientist, our PI. He is a clinical doctor, so he needs to do clinical trial in the future. So all of our study, we are designed for the clinical trial in the future. So I'm Jun-Yi and others are all the people have contributed to our project here. So give everyone a little bit basic concept of what is the focused ultrasound. So ultrasound is already FDA approved device that you can do the daily image about how was your organ development, is they okay or that they did not look normal. Recently, people try to use this, become the focused ultrasound to do some treatment so the energy can become more focused on certain area. You will not just broadly everywhere or exposed to the sun. So how sun can make energy to the tissue. So when you go to the music festival, the music was too loud. You can feel the ground is moving. So that's the mechanical force of the sound wave. So here, how we make the blood-brain barrier can be open is by the sound wave, that we only focus at the area we interesting about. So combined with the contrast agent that already approved by the FDA, that original is their daily routine imaging tool. So when the agent, it's actually it's microbubble, when they expose to the area have the sound wave, it will change the shape. And that change the shape will make it to your blood-brain barrier temporarily open and then release your drug or creatine into the brain area. So that's why we are going to do this and test whether we can apply our technique into creating a CTD disorder, and then how was the creatine can go into the brain. And we will use the cavitation feedback by how was the bubble change the shape and how they respond to the sound wave as an indicator how strong is this kind of the sound wave is strong enough or too strong. So currently, they have multiple clinical trials ongoing by focused ultrasound. It's ongoing, including the Alzheimer's disease, Parkinson, and the children brainstem tumor, and GBM and other epilepsy and ALS. They're all ongoing worldwide, everywhere with the clinical trial. And our lab is doing the brainstem tumor. Brainstem is control your heartbeat, your breathing, and it's very important area. Patient will not let your children to destroy that area. So how we can use our lab research by working on the animal, then can translate to clinical is by our design in the animal. We need to have a full detailed information, how was the drug chemical go into the brain before we can move to the clinical one. So that's why when we doing in the lab, everything, all the information is important for us to apply for FDA clinical trial. So previously, when we do the chemical drug, the molecular size can range from 300 molecular weight to antibody size. It also, we can deliver it into the brain area we are interested about. And this allows us to go to the clinical trial of 2 different trials. And now we have the device at the Children's National Hospital for tumor treatment. So based on this, we think the creatine, your daily supplement you are taking right now, should be able to deliver it into the brain. It's smaller than the chemical drug we are treat here. So here is we propose what we can do here. When we treat the animal model, the CTD have the creatine disorder, then we will treat the with the focused ultrasound in the brain. Then open the blood-brain barrier, then to see how was your brain uptake the creatine. So we will use the MRI scanning. So you will not sacrifice the animal. You will check, monitor how was their creatine level in the brain area. And then we will, because we do focused ultrasound, we need to confirm how much BBB, blood-brain barrier, has been opened. So it will also help us in the future how much or how frequently we need to open the blood-brain barrier for treatment. And also we use the brain integrity scan to make sure we did not harm the brain or damage the brain. And ultimately, we need to have all the family more care about behavior, the cognition one. We need to finish that before we can apply for the clinical trial. So here is our treatment cycle. We will scan the creatine at the baseline first. So we will check every individual mice and got their baseline creatine level. Then we notice that there are variation, even they are in the lab, and they are same company, same family. So it's very variation. And then we will, after we gather the creatine baseline level in the brain, we will start to treat the creatine daily water. Then we start to treat with the fast treatment for the BBB opening. And this treatment, we already have a solid day cycle right now in treatment. So now we are fixated. We will check once a week and every other day. The next day and second day, we will monitor the creatine peak, whether they are going up. And then at the same time, we will monitor whether the brain be damaged and how was the degree of the BBB opening on the animal. Then after cycling treatment of the fast treatment with the creatine supplement, then we will harvest the brain, sacrifice the animal to assay molecular change. So in this way, we can see how we can change the brain and how was the chemical change in the brain system. So, and then we will use some patient's fibroblasts. Your sample that have been donated, we are using it for our assay. And in the end, we will evaluate how was the behavior, cognition, or learning memory can be improved by our treatment. So now you already know the cycle we are doing here. So our baseline MRI scanning here, you can see in the wild type, the creatine peak is normal, close to the core line. And in the knockout animal, it's lower next to the core line peak. So our fast treatment is cover the whole brain area, but as a control, because we notice there is a big variation from individual to individual. So we decide to just make the animal have another site did not treat and as our control. And before we doing this, we already try whether we do this, the creatine diffuse to another site, or when we scan, does it change? And we solve that problem. So that's why we consistent in this way. And then the right side is our confirm of the BBB opening, some at the 24-hour and some at the 48-hour. We also have some 72-hour. You can continue monitor them. And all the animal, this is after second cycle of treatment. And the brain opening degree will be different than the first time. First time, everyone will be very synchronized in unique, very consistent way. Then after 3 cycles, we will harvest the brain. So here, after the converting of the BBB, we will check how was the brain integrity still be maintained. So like the first one, the wild type, they are all looking normal, no damage at all. And in the creatine knockout animal, they also look normal, not damaged by any fast treatment. But when we try to expand our population on the treatment, here each animal we all scan once of the image to check the brain integrity. We do notice some of the brain, they cannot sustain more than twice of the treatment. So you can see the third one at the second row have a damage already show up. Then we will stop treatment. They are not tolerant on this kind of treatment. So it's also related how we design in the clinical trial. So on why MRI is important for us is to monitor how much the treatment go into the brain and how long they can stay in the brain. And this is important for patient care about why we cannot see the cognition or brain improvement. So we want to make sure you are creatine to go to the brain first before we assay all the behavior. So here, the baseline, we already showed the low creatine. The low creatine is about 20% of the creatine relative to the chlorine peak. But when we measure the fast treatment after 24 hours, the creatine peak the maximum can reach to the 40% relative to the chlorine peak. So it has some improvement here. Then we also compare at the same brain, the left side and right side. So the black line here is the side we did not treat the fast treatment. the origin color is the one we have doing the BBB opening. So the body is exposed to the same creatine label. The only difference is the brain with the fast treatment to do the BBB opening. And we find that at the first fast treatment and second fast treatment, creatine do increase. And this creatine increase in the brain, it need to be quantified. Even we are just still using the eyeball to measure relative ratio to the colony, but we still need a lot of data process behind to remove the signal noise. Then we can give you the good quantification how much we improve in the brain. And that will be done by our collaborator at the Columbia. So here is our first batch of the creatine label in the brain. So you can see in the control part, we normalize to TAA in this case because the colon, after we doing the fitting, the post-process, their quality is not good enough as a control. So that's why we convert to TAA as a control because it did not change over time. So in relative, we can see creatine low is all lower level as what we are seeing in the pig. And in the knockout, it's relatively low. But when we try to compare the same brain, whether they are effective to each other. So in the wild type, the creatine level did not change too much in this kind of treatment. But when we treat in the creatine knockout animal, the baseline was low, the not untreated side is low. But when you're doing the BBB opening, the creatine do increase in the MRS peak. So you can see from the 20 to 50 or 30 to 40. So their range is different from animal to animal. So after we doing all the MRS quantification and analyze, we harvest the brain and want to make sure we do see it do uptake into the brain. So we harvest the brain, separate into two half. The left side collect as a fast treat control, and another side is their comparison control. So we also see the similar and match result that is similar to our MIS in vivo assay. So wild type has higher creatine, and in the knockout animal, their creatine is low. And after we compare the same brain on the control side and the fast treat side, all the creatine knockout animal has response to the fast treatment. All the individual animal that have the creatine peak increase when you are only treating one side of the brain. So this is to increase the creatine locally at the brain area. And this is similar. This LC mass is confirm our in vivo assay is on the same hint. So we also, after we harvest the brain to assay the phosphocreatine, we did not see too much change in the control side and the fast treatment side. And the creatine in the final product in the brain also did not have too much change. So we also assay other neurotransmitter, how was their change? So we noticed there is some neurotransmitter is being reduced, like the glutamate acid, dopamine, ergic transmitter, acetylcholine, and norepinephrine. And also we find some neurotransmitter is not changed at all after fast treatment with creatine supplement. So here we have the hint that the creatine supplement with the fast treatment might change brain activity, and the neurotransmitter seems to have an effect here. But we don't know whether the neuron circuit to change, the neuronal function to improve. So that's why we will need some behavior in the future. And people question about whether when you go to the brain, does it really go to the cell? So that's why we use the human cell here to do the fast treatment. It's a converted version of the focused ultrasound, try to use it in the tissue. So we call it a sonoporation. Like you treat the on the tissue, it will generate some temporary open of the brain, not brain, in the membrane of your cell. Then you will uptake the creatine outside of the cell. Then you harvest the cell to measure how was the fast treatment on the cell uptake of the creatine. So here we select multiple mutation from Colonial Institute. So your sample donate here with the history of your family, patient's onset of the day symptom. We select from here, and also the healthy control. So we use the deletion version of the patient's fibroblasts to test this one. We see the control is low of creatine, and fast only did not change too much. And you give the creatine only, the cell do have their own small capability to uptake the creatine. And when you do the fast with creatine supplement in the media, it do increase the creatine uptake much more here. But due to our sample size, we are not get significant yet, but we will continue work on that. And in the other side, the phosphocreatine, we did not see too much change here. So our ultimate goal here is to see whether the neuron do have improvement. So after we have know all the series treatment do have improved some, we see the evidence of the creatine go to the brain and the neurotransmitter change and whether their behavior can have improvement. So we will go work on this to help us to move on clinical trial. Then here is the short summary for everyone. We do see the safety of the fast treatment. If it's not tolerable, we cannot do that. So then that one, we will reduce the parameter or pressure in the future. And the safety and repeatedly on the animal, they are not damaged at all. They can sustain. So intracellular creatine also can be validated from our LC mass assay from we harvest the brain and also the cell fibroblast treatment assay. Now we are waiting for our behavior test to see how was our outcome before we can move to clinical trial. So then in the future, we will use the creatine with what you are treat your kids right now every day to do the clinical trial in the future. So here is our team, Frey Wu here. We got the idea from Dr. Berger, and so that's why we start this project. And the Sarah here is going to apply the PhD program. So if she applied to your school, please hire her. She worked with me on everything. So she knows how to breathing, how to do the MRIs, MRI, and including all the treatment, fast treatment on the animal. And we also cooperate with the Children's National and including the engineer part of the fast device. And also the GIAGO at the Columbia, their team is MRIs, they are MRI expert. So they will do many AI-based data post-process for us in the future to do more analyze. Yes, now here is an official slide. We are called for collaborator. Anyone have the new drug or new therapy you are ongoing, mRNA, protein therapy, anything, we are open to cooperate. If you already see it can improve in the animal with muscle improvement, but not in the brain, we are here for you to test whether it can improve, increase the efficacy in the brain. And yeah, thank you, everyone. Hire her.
Thank you for the interesting talk. I have a question. Maybe I wasn't sure on something that you were talking about towards the end. So could it be that the reason, if I understood correctly, you weren't detecting a change in activity is because you only opened the blood-brain barrier and the creatine is not in the neurons, but it's in the brain? Because you have to have the creatine transporter both in the blood-brain barrier and in the neurons themselves, the glia, the oligodendrocytes, to have creatine enter the cells. So is that what you were talking about with the sonoporation later on? It was a little unclear for
We
me.
do see, when we harvest the brain, the LC marks do show the creatine is increased here. So when we harvest the brain, the control side is not treated with FAS, and another side is treated with FAS. So we do see there have significant change on each animal. They do increase.
But is it in the cells? Is it in the Is it in the cells of the brain, or is it in the surrounding fluid?
It definitely will not be in fluid. So right side also have the fluid. Every one have the same fluid. They have the same blood. They have the same spinal ventricle area fluid.
But the focused ultrasound is only opening the blood-brain barrier. It's not opening the cell membranes of the neurons, the glia, and the oligodendrocytes, correct?
Yeah, so that's why we want to try on the sonoporation to make sure that part we can achievable. And also, we also try to find some dye to can validate whether we can mark the neuron, because we don't have a good tracker for the creatine label in fluorescence, so we might use some fluorescent tracker to do that.
Hi, my question is on like the practicality of this as a treatment for families. Like what are your thoughts on how often kids would need this therapy done, or what, like is any ultrasound provider able to do this, or would they need special training?
Spatial creatine? Oh, spatial training. So our focus ultrasound at the National Children's Hospital, they are ongoing treat the brainstem tumor. So they are training already right now for the brainstem. So for the other brain area, we need to approve by FDA, then we can try on the different brain area. But if brainstem, we can try it, then I think it should be safe. And how frequently in the patient will depend on our data in the future on the behavior part, because we don't know how it will be enough for patient to get that to improve in the brain.
Could you look at phosphocreatine to see that it's actually getting inside the cytosol based on Jerry's comments?
Phosphocreatine is here. After we harvest the brain, the phosphocreatine we do after the third fast, we harvest the tissue immediately. So it's in 30 minutes, we harvest the tissue right away, and including in the creatinine, we also measure it. And the GAA is not changed at all. So only total creatine is increased, but not at the phosphocreatine or creatinine and GAA.
So that argues that it's not getting in the cells.
It's quite variation, so we don't know. Yeah.
Okay, this is very non-scientific, but I know the blood-brain barrier, especially for CTD families, kind of a blessing and a curse because it still does protect the brain. Does your system only allow that creatine into the brain, or are we opening a door that other things could get in and cause infections and problems?
it could. That's a very scientific question, definitely. Yeah. So if you have some poor stuff in the brain, in the blood vessel, and it's already supposed to need to be diluted, secreted outside, but you have that high amount in your blood, then you will have a chance to get it. Yeah.
Thank you, Chinyi. On slide 12, I noticed that with the focused ultrasound treated data, it looked like the lipid measurement was very different. Do you have a theory around that? And did that return to normal after a time?
No, that we try a lot of time. We even try to scan multiple times and change the scanning model and frequency and change the time on the same individual. The lipid is always the same. The pattern is the same before we do any fast treatment. So their composition clear maybe have more or different in the area. Yeah, some even show has higher, only few cases show higher lipid on the left side, but most of them seems at the right side. Yeah.
Next up, I'd like to welcome Carmon Phillips. Carmon and her husband Warren live in New York with their 3 children, Elizabeth, Daniel, and Stephen. Apologies. Their daughter Liz was diagnosed with GAMT in December 2023 at the age of 29. They'd like to share a little bit about Elizabeth's stories to give hope to other families who are still searching for answers.
So having this on is very new, but thank you for having us. We are the Phillips family from Syracuse, New York, and our daughter was born in 1994 and was diagnosed with lactic acidosis secondary to mitochondrial myopathy around the age of 2. GAMT was discovered in 1994, but we would not find out that she had this until many, many years later. True failure to thrive. She only weighed 14 pounds at 2 years old. Global developmental delay, seizure disorder, low muscle tone, nonverbal. She didn't crawl until 2. She knee walked at 3. She started having grand mal seizures at 3. She had behavioral issues. She was self-injurious. The list goes on, as most parents and caregivers here can attest to. Since I only have about 5 minutes, I will jump to 2023, when at the age of 29, Liz had a medical crisis. In late June of 2023, her neurologist at the time had talked to us about redoing genetic testing to try and pinpoint the exact mitochondrial issue she had, so maybe we could treat her better, which we agreed to do. And at that same visit, he also started her on L-arginine and CoQ10. We started those supplements in July of 2023. On September 14th, 2023, she started having episodes which we would later realize were seizures. At that point, she had been seizure-free for over 10 years. By the end of September, she was having cluster seizures regularly, and by October, she was having a seizure every 20 to 30 seconds around the clock. We were in and out of the hospital, and she was dying, and nothing seemed to be helping her. Within 2 months, she lost 40 pounds. At her lowest weight, she was 78 pounds. She was bedridden and had lost the ability to do anything. She could barely sit up on her own. We had to carry her everywhere and move her from the bed to the couch every few hours to try and combat pneumonia. We had to turn her every few hours to try and prevent bed sores. We could count her ribs and see every vertebra. She was in so much pain. She was put on 3 different seizure medications that weren't working. It was a terrifying time in which we were very overwhelmed and felt completely helpless, as we did not know how to help her. At that time, we resigned ourselves that this was how mitochondrial disease ended. Mid-October, her genetic testing came back, and it was negative for mitochondrial disorder. We were shocked and confused. Our genetic doctor sent the testing back to do a broader search, and in November, it came back that she had GAMT. We had answers, and there was treatment. We realized then that the L-arginine was poisoning her. We immediately stopped giving that to her. Within a couple weeks, the seizures stopped. This began our journey to learn all we could about GAMT and what her life would look like now. We got into a pediatric specialist in our community who is actually in touch with Dr. Longo, and I'm sure has asked many questions on our behalf. And in February of 2024, she started creatine, ornithine, and sodium benzoate. We only did sodium benzoate for about 6 months, but we had to stop that because of negative side effects. We're here today to thank you doctors and scientists and specialists, because without you, we don't believe our daughter would be here today. You have been gifted with the ability to study and figure out rare and complex diseases, and we are forever grateful for the work that you do. Because of your work, our daughter has a new lease on life and is progressing far and beyond what we ever thought possible. Our specialist recently told us that when she first started treating Elizabeth, she was hopeful that we could get her back to her baseline and that she would be able to live a comfortable life. She never thought possible that Liz would surpass where she's been and continue to make progress and do things that she never did before. Almost 2.5 years into this new diagnosis, Elizabeth is like a new person. She has started using some sign language regularly and points or takes you to what she's wanting or needing, a skill that she did not have before. She's babbling like a toddler and trying to have back and forth conversations. She plays with her iPad. She uses her toy phone to try to take selfies. She dances to music. She hugs more often and is more intentional and laughs hysterically at jokes, which is good because we are a super sarcastic family, and she also is. She is so fun to be around, and we are forever grateful that this disorder was finally found. Today, she does struggle with uncontrollable tongue movement and swallowing dysphagia, and we wonder if it's due to the damage done from the L-arginine. But we are starting to see progress in so many other areas. We're hopeful that this will continue improving as well. We also have had some small setbacks with seizures, which can be scary, but we're learning more about the ornithine and those levels and things like that. We don't have all the answers about GAMT and what she will become or how this disorder will progress, but I will tell you that without the work done to find this disorder and the protocol to help children and adults with this disorder, her story would have ended a long time ago, long before it should have. So thank you for your continued efforts, and please keep working and digging to find cures and treatments so kids like ours can have a fulfilling life. Thank you.